Welcome back to the deep dive. You know, you see it on TV all the time, the detective with the black light. But today we're going past that. We want to figure out how forensic scientists actually prove what they find is, solid evidence for court.
Exactly. We're diving into the science of identifying semen in forensic samples. It's really this layered process based on specificity.
Right. So we're using Richard Lye's forensic biology as our guide here, kind of mapping out how they go from a maybe to a definite yes.
That's the plan. We'll look at the quick screening tests, first finding something, and then dig into the methods that absolutely confirm it's semen, the kind of proof you need legally.
Okay. Yeah, sounds good. Let's follow that evidence trail. Where do we start? What exactly is semen from a forensic perspective?
Right. So when we talk about semen in forensics, we're talking about this complex biological fluid.
And not just one thing, then.
No, definitely not. A typical ejaculate, it's actually a pretty small volume, maybe 2 to 5 milliliters, but it's got two main components. You've got the seminal fluid, that's basically the liquid part, secretions from different glands, and then you have the spermatozoa, the actual sperm cells.
And I understand the number of sperm cells can be huge.
Oh, absolutely staggering. You could be looking at anywhere from 10 million to like 100 million per milliliter. It's a huge number.
Wow. And that high count must be useful forensically.
It really is. It means that even in tiny stains or older stains, there's often still enough cellular material to potentially get DNA later on.
Okay, but you mentioned the fluid part, the secretions. Are there important clues in just the liquid itself?
Definitely. Those secretions contain key chemical markers that we rely on, especially for the initial tests, even before we confirm sperm cells are present.
So where do these secretions come from?
Well, about 60% of the seminal fluid comes from the seminal vesicles. And this fluid contains a really important component called flavin.
Flavin, is that the black light thing?
That's the one. Flavin is what causes semen to fluoresce under certain wavelengths of light, usually UV or blue light. That's the basis for the alternate light source screening.
Okay, got it. What else is in that seminal vesicle fluid?
It also has proteins that help the semen coagulate initially. The main one we look for sometimes is seminal vesicle-specific antigen, or SVSA. You might also hear it called Semenogelin, or SG.
Age. Okay. And what about the other glands? You said 60% from seminal vesicles.
Right, about 30% comes from the prostate gland. And the prostatic fluid is crucial because it contains really high concentrations of two key forensic markers.
Which are?
First, there's an enzyme called acid phosphatase, AP for short. It's vital for presumptive testing.
AP, okay. And the second?
The second is prostate-specific antigen, or PSA. Sometimes it's called P30. These two, AP and PSA, are really the workhorses because their letters are just so incredibly high in prostatic fluid compared to other body fluids.
Makes sense. High concentration means easier detection. Now, what about the sperm cell itself? If you actually find one, what are you looking for under the microscope?
Structurally, a spermatozoa has three main parts that are pretty distinct. There's the head, which is critical because it holds the nucleus. That's where the DNA is packed.
Right, the genetic material.
Exactly. The head also has this structure called the acrosomal cap, which contains enzymes needed for fertilization, though that's less critical for forensics. Then there's the middle piece.
What's in there?
That's packed with mitochondria, which basically provide the energy for the tail. And finally, you have the tail, the flagellum, which is responsible for motility, for movement.
And these cells are pretty unique, biologically speaking.
Yeah, they are. Mature sperm are highly specialized. They actually lack a lot of the standard organelles you find in other cells, like the Golgi apparatus. That unique structure helps differentiate them visually from, say, epithelial cells that might be mixed in the sample.
That's important. But what if someone has had a vasectomy or has a condition where they don't produce sperm? Does that mean no evidence?
Ah, good question. No, not necessarily. This is a really important point. Even with a vasectomy, where the tubes carrying sperm are blocked, or in conditions like azoospermia, meaning zero sperm count,
Right.
the person still produces seminal fluid. So all those chemical markers we talked about, AP from the prostate, PSA, even SJAG from the seminal vesicles, they're still present. They're still detectable.
So you can still identify the fluid as semen.
Absolutely. Now, you won't find sperm DNA in those cases, obviously. But interestingly, you can sometimes still get a DNA profile from epithelial cells, just skin cells, basically, that are naturally shed into the seminal fluid during ejaculation.
So the absence of sperm doesn't automatically mean you hit a dead end.
Correct. It just changes what you're looking for and what type of DNA you might recover.
Now, you mentioned these markers. How long do they actually last? Does evidence degrade? That seems critical for investigations.
Oh, hugely critical. Marker stability dictates everything. Some markers are much more robust than others.
Like which one? Which is the most stable?
PSA is pretty remarkable. In a dried stain, kept at room temperature, PSA has a half-life of about 3 years.
3 years. Wow.
Yeah, it holds up really well. That's why it's often detectable in older evidence, cold cases.
And what about AP, the other big one?
Acid phosphatase activity, well, that's much less stable. Its half-life is significantly shorter, maybe around 6 months, even under ideal conditions,
Right.
like 37 degrees Celsius. And if the stain gets wet or stays damp, AP activity drops off much, much faster.
So knowing the age and condition of the stain immediately tells the lab which tests are likely to work?
Exactly. It guides the whole testing strategy. You wouldn't rely heavily on an AT test for a really old, degraded stain, but PSA might still be a viable target.
Okay, that biology background is super helpful. Let's move into the lab now. Section 2. Finding the stain. These are the presumptive tests, right? The initial search.
Direct, presumptive assays. Think of it like a first pass. They're usually fast, sensitive, but not absolutely specific. The goal here is just to locate potential areas of interest.
And this is where the famous blacklight comes in, the alternate light source, or ALS.
Yep, that's often the very first step. We use specific wavelengths of light, usually in the blue-green range, around 450 to 495 nanometers.
Okay.
This light excites the flavin molecules in the seminal fluid, remember? And that causes the stain to fluoresce, to glow, usually a yellowish-green color when you look at it through orange goggles or filters.
Why the orange goggles?
They filter out the blue light from the source itself, making the fluorescence from the stain stand out much more clearly. It's great for scanning large items quickly, like bedding or clothing, without damaging anything.
And this seems like a big but you said flavins make it glow. Do only semen stains glow like that?
No, and that's the crucial caveat. ALS is highly sensitive, but it's not specific. Lots of other biological fluids can fluoresce too. Saliva, urine, sometimes even vaginal secretions, although often less intense.
Yeah, non-biological things.
Oh yeah. Detergent residues, certain fibers, food stains. Lots of things can glow under an ALS. So a positive fluorescence only suggests the possible presence of semen. It tells you where to look next.
It's just a pointer, not proof.
Exactly. It narrows down the search area. Once you have a fluorescent spot, you move to the next step, a presumptive chemical test.
And that would be the acid phosphatase AP test.
That's the main one. We know AP is present in really high concentrations in prostatic fluid, much higher than in most other body fluids. So we target that enzyme activity.
How does that work? I picture the swab test from TV where it turns purple instantly.
That's pretty much it. It's a colorimetric assay. The most common method uses a chemical substrate called anafyl phosphate.
Okay.
So you moisten a swab, gently rub the suspected stain area, and then add a drop of the substrate solution and a second chemical, usually something called fast blue B.
Fast blue B.
Right, it's a stabilized diazonium salt. Now, if AP is present at high levels, it acts like chemical scissors. It quickly chops a phosphate group off the naphthyl phosphate substrate.
And that reaction product.
That product immediately reacts with the fast blue B to form a vibrant purple azo dye.
Ah, so the purple color means AP is present.
Yes, but the key is the speed. Because seminal fluid has so much AP, this reaction should happen very quickly, usually within 60 seconds. A rapid, strong purple color change is a strong indicator for semen.
Okay, but you mentioned AP isn't unique to semen. It's found in other places, like vaginal secretions, right? How do labs avoid getting false positives all the time, especially in sexual assault cases where fluids might be mixed?
Good point. Specificity is definitely a concern. Labs improve it in a couple of ways. First, using anaphyl phosphate as the substrate is actually better than some older substrates because prostatic AP reacts with it particularly well.
Okay.
But the more important technique is using inhibitors. They can add tartrate ions, usually L-tartrate. Tartrate specifically inhibits the activity of AP from sources other than the prostate, like vaginal AP.
So if you add tartrate and the purple reaction still happens quickly?
Then you can be much more confident that the AP activity you're seeing is actually from prostatic fluid, and therefore likely semen. It adds another layer of specificity to the presumptive test.
That's clever. Now, you also mentioned mapping stains on large items like a whole shirt or sheet. How do they do that without cutting purple spots all over it?
For that, they often use a slightly different, even more sensitive AP test, a fluorometric assay, commonly called the MUP test.
MUP.
Stands for 4-methylmbilipherone phosphate. Instead of producing a color, when AP acts on MUP, it removes the phosphate group and creates a product that fluoresces really intensely under UV light.
Ah, so it glows instead of turning purple.
Exactly. So you take a large piece of filter paper, dampen it slightly, and press it firmly against the garment or bedding. This transfers traces of any seminal fluid onto the paper.
Like a blotter.
Precisely. Then you spray the MUP reagent onto the filter paper, wait a few minutes, and view it under UV light. Any areas where AP was present will glow brightly, showing you the exact location and shape of the original stain pattern on the evidence.
That lets you see the whole picture without damaging the item itself too much.
Right. You can then just cut out the specific glowing areas from the original evidence for the next stage, the confirmatory tests.
Okay, so we found potential stains with light, and we've got a strong chemical hint with AP. Now we need the definitive proof, right? Section 3, confirmatory assays. What's the absolute gold standard?
The absolute #1 definitive proof that a stain contains semen is the microscopic identification of spermatozoa. If you see sperm cells, you know it's semen, period.
Makes sense. But you mentioned earlier that samples can be mixed with other cells, like epithelial cells from the victim. How do scientists tell them apart clearly under the microscope?
They use a special staining technique, a differential stain. The most common one is called the Christmas tree stain.
Christmas tree stain. Why that name?
Because of the colors. It uses 2 different dyes. The first is nuclear fast red, NFR. This stains the nucleus inside the sperm head and also the acrosomal cap, a bright pinkish red.
Okay, redheads.
Exactly. The second dye is picro-indigo carmine, PIC. This stains the sperm's neck and tail a contrasting blue-green color.
Ah, so redhead blue-green tail, like a little Christmas ornament.
Sort of, yeah. And the key is that other cells, like vaginal epithelial cells, stain differently. Their cytoplasm might pick up the blue-green PIC stain, but their nuclei stain red with NFR. So the sperm cells stand out very clearly with their distinct red heads and blue-green tails against the other cellular background.
That visual confirmation sounds pretty undeniable.
It is. Seeing those characteristic stained sperm cells is considered conclusive proof of semen.
Now, for DNA analysis, getting a clean sample of just the suspect's sperm DNA, separate from the victim's cells, is crucial. How do they physically isolate just the sperm cells if they're all mixed together on the slide?
That's where some really cool technology comes in, particularly laser capture microdissection, or LCM.
Laser capture sounds like sci-fi.
They're pretty amazing. After they've stained the slide and identified the sperm cells visually, they use a very precise, computer-controlled infrared laser. The laser is aimed directly at a single specific sperm cell, or a small group of them. Above the slide, there's a special cap coated with a thermosensitive polymer film.
Thermosensitive. Melts with heat.
Exactly. The laser pulse briefly mills a tiny spot on that polymer film directly above the targeted sperm cell. The melted polymer touches the sperm cell and instantly solidifies again, sticking to it.
So it, grabs the cell.
Precisely. Then you just lift the cap away, and it plucks only the targeted sperm cells right off the slide, leaving the other cells behind.
Wow. So you get a super clean sample of just the cells you want for DNA testing.
Exactly. It's incredibly precise and avoids a lot of the challenges of traditional chemical methods used to separate sperm and non-sperm DNA fractions, especially in difficult mixed samples.
That's really impressive tech. Okay, so microscopy is gold standard #1. What if you can't find intact sperm cells, maybe due to degradation or azoospermia? What's the next confirmatory route?
Then we move to identifying those specific protein markers we talked about earlier, using immunological methods. The 2 main targets are PSA and Semenogelin, SG.
Let's start with PSA. How do they confirm its presence definitively?
There are a few lab techniques like ELISA, but very commonly, especially for initial confirmation, they use immunochromatographic assays. These are often formatted as simple test strips or cassettes, like the ABA card PSA test, for example.
Like a pregnancy test almost. How does it work?
Very similar principle, actually. You extract the sample from the stain, put a few drops onto the test device. Inside the strip, there are antibodies against human PSA that have a colored label attached, usually pink or red.
Okay.
If PSA is present in your sample extract, it binds to these labeled antibodies, forming a complex. This whole complex then travels up the strip by capillary action.
Like liquid moving up paper towel.
Exactly. It reaches a specific area called the test zone, T. In this zone, there are other anti-PSA antibodies that are immobilized, stuck to the strip.
Okay, like a trap.
Right. These immobilized antibodies capture the PSA-labeled antibody complex as it flows past. This concentrates the colored label in that zone, forming a visible pink line. If you see a line at T, it means PSA was detected.
And there's usually a control line too, right?
Yes. Further up the strip is the control zone.
Yeah.
See, this has antibodies that capture any excess labeled antibody, regardless of whether PSA was present. A line must appear at C to show the test worked correctly. So 2 lines, C and T, is positive for PSA. 1 line, C only, is negative.
Seems straightforward, but you mentioned earlier something called the high-dose hook effect. What's that about? Sounds like it could cause problems.
It can. It's a potential pitfall with these types of tests. It happens when you have a massive amount of the target antigen, in this case, PSA, in your sample, way more than the test is designed for.
So too much evidence is bad.
Well, think of the labeled antibodies and the capture antibodies in the test zone, like dance partners needing to link up via the PSA molecule. If there's an overwhelming flood of PSA, all the binding sites on both the labeled antibodies and the immobilized capture antibodies get completely saturated just by free PSA molecules floating around.
Ah, so they can't form the sandwich needed to hold the color label in the test zone?
Exactly. The labeled antibodies might bind PSA, but they can't then bind to the capture zone because it's already blocked by other PSA molecules. So the labeled antibodies just wash past the test zone without being trapped.
Lead into no line at T.
Correct. No line at T, even though the sample is actually loaded with PSA, it gives you a false negative result purely because the concentration was too high.
How do labs handle that?
It's usually pretty simple if they suspect a hook effect, maybe based on a very strong AP result or the nature of the stain. They just dilute the sample extract significantly, maybe 1 in 100 or even more, and run the test again. Diluting it brings the PSA concentration back into the optimal range for the test to work properly.
Okay, that makes sense. Dilute and retest. Now, besides PSA, you mentioned Semenogelin, SG, or SVSA. Why is testing for SHE becoming more common?
There are a couple of really good reasons. First, specificity. While PSA is overwhelmingly found in semen, trace amounts can sometimes be detected in other fluids like urine or even breast milk in rare cases. Semenogelin, on the other hand, is considered much more specific to seminal fluid. It's generally not found in those other fluids.
So potentially fewer false positives or ambiguous results?
Potentially, yes. The second big advantage is concentration. SHE's is involved in that initial clotting of semen, remember. Its concentration in seminal fluid is actually much, much higher than PSA's concentration.
Higher concentration means easier detection, especially in trace amounts.
Exactly. So kits designed to detect seed, like the RSID semen test, which uses immunochromatography similar to the PSA test, can be very sensitive and highly specific, making them a preferred choice in many labs now, either alongside or instead of PSA testing.
Okay, so microscopy, PSA testing, SG testing. What's the third major confirmatory approach? You mentioned something cutting edge.
Right. The most modern approach, and arguably the most specific biologically, involves looking not at the proteins, but at the RNA molecules that code for them.
RNA, like DNA's cousin.
Sort of. Specifically, we're looking for messenger RNA, or mRNA. These are the temporary blueprint copies made from DNA genes, which tell the cell's machinery what proteins to build.
So instead of finding the final product, like PSA protein, you're finding the instruction manual to build PSA.
That's a great analogy. Using a technique called reverse transcriptase polymerase chain reaction, or RT-PCR, we can detect specific mRNA sequences that are known to be expressed only in cells relevant to semen production, like cells in the prostate making PSA mRNA, the gene is KLK3, or even mRNA specific to sperm cells themselves, like protamine-1 or protamine-2 mRNA.
And the advantage is?
Extreme specificity. If you detect mRNA for protamine-1, for example, you know with very high certainty that sperm cells were present, even if they were too degraded to see under a microscope. It ties the finding directly to the specific cell type or gland of origin. Plus, these methods can often be automated.
Sounds powerful. What's the catch? Why isn't everyone just doing RNA testing all the time?
The big limitation is stability. RNA, unfortunately, is much more fragile and easily degraded than proteins like PSA or SG or even DNA. It gets broken down quickly by enzymes called ribonucleases, which are pretty much everywhere.
So it doesn't last as long in evidence?
Generally, no. While RNA detection can be fantastic for relatively fresh samples or well-preserved evidence, it's much less likely to be successful on old, dried stains or samples that have been exposed to harsh environmental conditions. The RNA just might not survive.
So it's a trade-off. Incredible specificity, but only really viable for certain types of evidence.
Pretty much. It's a very powerful tool in the toolbox, but maybe not the first choice for a decades-old cold case, whereas PSA might still be detectable.
Okay, that gives us a really clear picture of the whole sequence, from finding a glow to proving it at the molecular level. you had to boil it down. What's the main takeaway from this whole forensic identification process?
I think the key thing to understand is that it's a hierarchy of testing. You start broad and sensitive using light, then basic chemistry like the AP test. These help you find potential evidence quickly.
The screening phase.
Right. Then, for anything that screens positive, you move up the ladder to tests that are progressively more specific, but maybe slower or more resource-intensive microscopy. Specific antigen tests like PSA or SG, and finally, RNA. Each step narrows down the possibilities until you reach definitive, court-admissible proof.
It's a funnel, basically. Start wide and narrow and certain.
That's a good way to put it. It's all about managing sensitivity versus specificity at each stage.
And thinking about that whole process, what really strikes me is how much the success of an investigation, especially an older one, hinges on the basic chemistry and stability of these markers.
Absolutely. That difference between PSA lasting for years, while AP or RNA degrade much faster. That's not just lab trivia. That directly impacts whether a case from 5, 10, even 20 years ago might still be solvable today. The inherent stability of the biological evidence sets the timeline.
It determines what's possible.
It really does. The chemistry dictates the limits of the investigation in many ways.
So maybe a final thought for our listeners to consider. Where does this go next? How might technology change the stability equation in the future?
That's a fascinating question, isn't it? Will future breakthroughs focus on, say, developing methods to protect fragile RNA molecules from degradation at a crime scene? Or maybe biochemists will discover even more stable protein markers, unique to semen, that can persist in the environment for even longer than PSA. Extending that window for justice even further, that's likely where a lot of research is heading.
Finding ways to make time matter less when it comes to evidence. That's definitely something to think about. Thank you so much for breaking all that down for us today. This has been a really insightful deep dive.